লাস্ট কন্ট্রিবিউশন স্যার রেখেছিলেন, তারপরে ডক্টর গোলাম ভট্টাচার্য স্যার এসেছিলেন, ওনারা টক্স দিয়েছেন। যেহেতু এটা আমাদের সেকেন্ড ডে স্যার, তো আর আমাদের প্রথম এটা প্রয়াস ছিল সায়েন্স টেস্টে, তো আমরা সেকেন্ড ডেতে আপনার টক্স আমরা স্যারের টক লিখেছি, মানে ডক্টর সমিত স্যার আমাদেরই অর্গানাইজেশন সেকশনে, তো আমি স্যারকে জানাই তো স্যারের টপিকটা দ্য জর্নি অফ মলিকুলার বেসিস স্ট্রাকচার, সবাই দেখতে পাচ্ছেন স্যার। তো স্যার
thank you very much sir for your kind introduction. At first I would like to thank the organizing committee members of Miracurall25 and the Science Faculty Students Union for the invitation and giving me the opportunity to interact with the students as well as Science Faculty members. So good afternoon everybody. Today the title of my presentation is A Journey with Molecular Machines. A molecular machine is an assemblage of parts that produce quasi-mechanical movement in response to specific stimuli. Many biological molecules can perform useful work. So this is membrane fusion machinery. Membrane fusion is a fundamental process in life and plays a key role in exocytosis, endocytosis, intercellular trafficking, envelope virus infection, fertilization and so on. However, spontaneous membrane fusion cannot be achieved due to two reasons: electrostatic and steric crowding between the two membrane surfaces is the primary reason. When two bilayers are very close with respect to each other, almost 2 nanometers apart, a much stronger barrier is encountered due to the layer of water molecules which are structurally aligned between the two membrane surfaces. So you have to break the structure of water molecules to get membrane fusion. In biology, these energy barriers are believed to be overcome in the presence of a snare protein, soluble A-methylvalimide sensitive factor attachment protein receptor. Snare link the two membranes and drag them together in close proximity, and two separate lipid membranes can form a single continuous bilayer. So in 2013, Professor James Rothman, Randy Seckman and Thomas Sudor received the Nobel Prize for their discoveries of machinery regulating vegetable traffic, a major transport system in our body. And Professor James Rothman is the father of snare hypothesis. So a lot of neuronal cells are present in our brain. So when action potential arrives at the nerve terminal, then voltage gated calcium channels are open. The neurotransmitter releases through membrane fusion. You can see neurotransmitter releases through membrane fusion, particularly snare protein mediated membrane fusion. At first it forms thermodynamically stable 4 helix bundles, then hemifusion, then full membrane fusion. Then the 6 snare complexes are subsequently disassembled for reactivation, and this reaction is going to a cyclic manner where snare protein acts as a catalyst. বাংলা একটু বলি যে ঘুমের পরে যদি মশা কামড়ায় আমরা কিন্তু দেখছি না, হাত কামড়ানো মানে চলে যাচ্ছে, মানে ব্রেন তোমাকে ইনফরমেশন দিচ্ছে। এর জন্য অনেক প্রোটিন দায়ী, তার মধ্যে একটা প্রোটিন হচ্ছে স্ন্যাপ, অ্যাকশন পোটেনশিয়াল একটা কারণ, ঠিক আছে? So chemists are very much interested to develop artificial membrane fusion machinery. So you can change this recognition protein, you can use hydrogen bonding type interaction, cyanuric acid and melamine type interaction, you can use covalent bond formation, boronic acid and cis diol, you can utilize complementary DNA strand, you can use complementary DNA strand, you can also use complementary peptide strand. This type of membrane fusion machinery can be utilized for artificial targeted drug delivery. So in case of corona, so COVID-19, you know, SARS-CoV-2, there is a spike protein, right? And another receptor is ACE2 receptor. So they can recognize and membrane fusion take place and virus can enter into the cell. This is also a membrane fusion. So another important molecular machine is RNA polymerase. RNA polymerase is an enzyme which can synthesize RNA using DNA as a template. Here you can see this is RNA polymerase. Unwinding the DNA double helix, and one helix strand is utilized to synthesize mRNA, messenger RNA, which is really important. And the next step, messenger RNA will synthesize protein in our body. So Roger T. Conrad received the Nobel Prize in 2006 in chemistry for creating detailed molecular images of RNA polymerase during various stages of the transcription process. Another important biological molecular machine is kinesin or dynein-1. It can move along the microtubule using ATP as a chemical fuel of biological system. So many biological molecules can perform useful work such as kinesin and dynein. আমাদের বডিতে আমরা যখন হাঁটছি আমাদের শরীরের মধ্যে কাইনেসিনও হাঁটছে, তার উপরে হাঁটছে মাইক্রোটিউবুলের উপর। এনার্জি কে পাচ্ছে, এটা ইম্পর্টেন্ট পার্ট। কে এনার্জি কি করে পায়? ফুয়েলটা কি? এখানে ফুয়েলটাকে ঠিক ATP as a fuel. So another important biological molecular machine is F1 F0 ATP synthesis. That is important. So in our body we have F1 F0 ATP synthesis. কোথায় থাকে? Mitochondria. Very good. Mitochondria, inner mitochondrial membrane, which can produce ATP, right? So it is actually two rotary motor and a common axis. People may have mimicked this F1 F0 ATP synthase. So this is the man-made motor actually. And this is ribosome. Ribosome is a macromolecular machine which can synthesize protein using mRNA as a template. So you can see this is the ribosome and this is the tRNA, transfer RNA. It will bring the amino acid building block. And now mRNA will act as a template and it will synthesize protein or peptide in our body. This is nicely done by nature. In our lab, we use nasty chemicals, DNA, coupling reagent, but see how nature creates protein or peptide in our body without using this type of nasty reagents. And then see, peptide or protein chains are growing. Now, this biological molecular machines is really complex. So chemists have mimicked this type of molecular machine and developed some mechanically interlocked molecules. So, how you will assemble the parts of molecular machine? You can use non-covalent interactions like hydrogen bonding, electrostatic interaction, halogen bonding, aromatic donor acceptor interaction. You can use also covalent bond formation by using copper catalyzed link chemistry as well as copper link chemistry. And you need also the fuel to run the machine also, right? So in 2016, Professor James C. Professor John Kersobat, Sir Fraser Stoddart and Ben Beringa received the Nobel Prize in chemistry for the design and synthesis of molecular machines. And he is the father of Rotaxane, Sir Fraser Stoddart. So this is interlocked molecules. This is interlocked molecules, catenanes. And this one is Rotaxane, right? So catenane developed by John Pierre Sauvage, Rotaxane mainly developed by Fraser Stoddart, and Ben Beringa made molecular rotite, right? And nowadays you can see in computer chip Rotaxanes are there. So what is mechanical bond? Since there is no covalent bond between the components of catenane and rotaxane, the linkage has been called mechanical bond, right? So this type of mechanical architecture, mechanical bond like architecture, you can see in everyday life. From ornaments, you can see ornaments, chains, and this is baby's toys, and this is the Olympic logo. You can see even in the road also, right? So this is the 4 catenane. And you can see baby's toys as well as bird toys. This type of rotaxanes are also there. So in nature also, this type of mechanical bond is there. So particularly in protein, virus, so they are using not special type of knot, molecular 8, and this is catenane based on DNA, and this is the important peptide based compound which is known as lysopeptide, mycosin J25. This is also cysteine knot peptide. Nowadays this is very playing important role as an antibacterial agent. Recently published on Nature paper 2025 based on this lysopeptide. So precious product develop a rotaxane. This is actually like a cable car or molecular switch. You can see interesting undergraduate level chemistry. So this is what? This one is benzene, right? And this one biphenyl system. Between these two, this one is more electron rich. So this blue box is electron deficient. Now blue box will stay on top of this benzene ring. Now if you protonate benzene, what will happen? Nitrogen will be protonated. Then it will form NH2 plus. Then due to electrostatic repulsion with 4 positive charge of this blue box, this ring will move to here in presence of acid, right? Now if you put base, what will happen? This ring will move here. So this is like a cable car or like a molecular switch, right? Based on simple, the fuel is here, acid base, right? pH. And simply you can use electron rich molecule and electron deficient molecule to form non-covalent type interaction. Further, they have developed this rotaxane based nanobombs. They have used silica mesopress materials, and then what they did, they have incorporated some drug molecules and they put some rotaxanes here. So when the macrocycle is sitting here, then the valve is blocked. Drug will not come out. Now in presence of acid, base or light, the macrocycle will come in the station 2. Now the valve is open, so the drug will release. Now if you want to close the valve, again if you put acid or if you put light, then the valve will block. So this is highly selective rotaxane you can use for the controlled drug delivery. So this is another molecular machines. This is called peptide synthesizer. What they use? They use one rotaxane molecules. They have attached reactive arm at the rotaxane. See, and then you can see removing building blocks, and it will form peptides. Like nature did, they have synthesized peptide by using this type of rotaxanes. And then there is no stopper, it will come out and you can get peptide the protein from this rotaxane template. So when I started my independent research career as a synthetic organic chemist, the very first question came into my mind: how to utilize synthetic organic chemistry to learn biology. Initially I was curious to know why does the cells have so many organelles like cytoplasm, nucleus, mitochondria, Golgi apparatus, endoplasmic reticulum, and so on. Then we emphasize that each organelle is actually a small reaction vessel. It is not what we call. Each organelle is actually a small reaction vessel. So, in endoplasmic reticulum, small amino acids are being stitched up to form protein. In lysosome, which is known as suicidal bags of the cells, where proteins are broken down into small amino acids, right? Nucleic acids are broken down into small building blocks, right? In Golgi, proteins are glycosylated. In peroxidome, long lipid chains are peroxidized into small lipid chains. Among the various cellular organelles, mitochondria are an important target for all cells. Mitochondria are the powerhouse of cells and control cellular function and fate. Mitochondria play a key role in programmed cell death, such as apoptosis. Mitochondrial dysfunction are associated with a range of neurodegenerative and metabolic disorder like Alzheimer, Parkinson, obesity, diabetes, and cardiovascular diseases. So mitochondrial targeting and imaging are the key steps for the design of drug molecules. However, it is very difficult to target mitochondria. Now, question is why it is very difficult to target mitochondria. So you can see this is the typical mitochondria. It has outer membrane, inner membrane, intermembrane space, cristae, and mitochondria has its own DNA. এটা একটু বলে রাখতে চাই ছাত্র আছো। আমাদের শরীরে দু জায়গায় ডিএনএ পাওয়া যায়। একটা হচ্ছে নিউক্লিয়াস আর একটা হচ্ছে মাইটোকন্ড্রিয়া। নিউক্লিয়াসে ডিএনএটা থাকে তাতে বাবা মা দুজনেরই বৈশিষ্ট্য থাকে। কিন্তু ইন্টারেস্টিং মাইটোকন্ড্রিয়াতে যে ডিএনএ থাকে মায়ের থাকে বৈশিষ্ট্য। এখানে কিন্তু বাবার বৈশিষ্ট্য নেই। অনলি মা। সো মাদারের ডিএনএ একমাত্র ভাবে হচ্ছে তুমি মাইটোকন্ড্রিয়া। ঠিক আছে? So this is important part. So why it is very difficult to target mitochondria? Because it's a unique cellular organism. Double layer membrane. Another important part is inner mitochondrial membrane potential, which is highly negative, minus 150 to minus 180 millivolt for normal cell, and for cancerous cell it is even higher, minus 220 millivolt. It is really unique. এরকম কিন্তু আমাদের শরীরে আর কোথাও নেই। So it offers a design opportunity to target mitochondria. Now question is why it is highly electronegative? Inner mitochondrial potential is highly electronegative. Why? Because it is due to the electron transport chain. তোমরা এটা দেখেছো, electron transport chain. Another important part is a lipid called cardiolipid. It is negatively charged. That is why inner mitochondrial potential is highly negative. So we are particularly interested to target inner mitochondrial membrane potential. So we have designed and constructed lipophilic cationic triphenyl phosphonium group to target mitochondria. We are also interested to design and synthesize near infrared chromophores. Why we are interested about near infrared chromophores? Because in our body we have peptide, protein, nucleic acid. This type of molecule absorbs and emits in the UV region. We have protein, peptide, this type of molecules, nucleic acid, this type of molecule, right? It looks like absorb for UV. We have melanin, hemoglobin, oxyhemoglobin. This type of molecule absorbs and emit in the visible region. And we have water in our body. Water absorbs in the IR region. So in our body we don't have any chromophore that absorbs and emit in the near infrared window. It is around 650 to 900 nanometer. Now it is 650 to 1000 nanometer for near infrared 1 and 1000 to 1700 for near infrared 2. People are working on near infrared 2 chromophore. So we are interested about squaring die, because so far squaring die has been overlooked to target mitochondria. Now, we hypothesize that the highly electron deficient squaring chromophore is vulnerable towards the nucleophilic attack by the exposed cysteine of mitochondrial proteins as well as mitochondrial glutathione. Right? And these are crystal structures of this molecule. And the counter analysis PFC minus. So we have performed confocal laser scanning microscopic assay using epithelioid surface carcinoma HeLa cell line, and this is the commercially available dye. You can see good signal from the mitochondria. But for our dye, we observe very weak signal. To understand the mechanism, we have performed fluorescence titration assay using glutathione and 2-mercaptoethanol SH containing compound at mitochondrial pH. Mitochondrial pH is very important. আমাদের শরীরে কিন্তু বিভিন্ন জায়গায় বিভিন্ন pH, তাই তো? লাইসোজোমের pH কত? 4 থেকে 5, এন্ডোজোম 5 থেকে 6, মাইটোকন্ড্রিয়া 8। ঠিক আছে, মাইটোকন্ড্রিয়া এটা বেশি। ঠিক আছে? So we have performed this titration using glutathione as well as 2-mercaptoethanol at mitochondrial pH, and we observe bleaching of the dye. To understand this mechanism, we have performed molecular docking assay using mitochondrial ADP synthase, and we have seen that exposed cysteine is very close to the squarene chromophore, and it is interacting with the squarene chromophore. So that is why we observe bleaching of the dye inside the mitochondria. Now question is how we will solve this problem. Now if you take this dye and put it into a molecular container, then you can protect the dye from nucleophilic attack. So this is called rotaxane. Now question is how you will synthesize rotaxane. সুচে সুতো ভরা, তাই না? সুচে সুতো করছি, সুতোটা বেরিয়ে আসছে, তাহলে কি করব? দুপাশে গ্রিড দিয়ে দেব। সেইটাই হচ্ছে কনসেপ্ট রোটাক্সেন। So actually this is threading and then stoppering. Another way you can synthesize template directed clipping method. This is called template directed clipping method. You can synthesize rotaxane. You can also use slipping method to synthesize rotaxane molecules. So we are particularly interested about template directed clipping method to synthesize rotaxane molecules. So we have synthesized this type of rotaxane molecule using solid phase synthesis protocol on 2-fluorobutyl chloride resin, and here we have incorporated macrocycle. We hypothesize that the macrocycle will provide the center and protect the chromophore from nucleophilic attack inside the mitochondria. So we are lucky to have the crystal structure of this rotaxane molecule. Here you can see this is stabilized through hydrogen bonding as well as donor acceptor donor type interaction. This anthracene is donor, squarene part acceptor, and this is donor. And donor to acceptor distance is around 3.7 angstrom. And this is the three-dimensional crystal structure. You can see this is the interlocked molecules and stabilized through donor acceptor donor type interaction. This molecule is non-cytotoxic towards both the cancerous as well as non-cancerous cell line. And now we have performed glutathione assay. You can see this compound is stable towards glutathione. Now glutathione attack is not possible because macrocycle is surrounded, right? So we have performed confocal laser scanning microscopic assay using epithelioid surface carcinoma HeLa cell line, and we have observed nice colocalization inside the mitochondria. And this is a controlled dye lacking the lipophilic cationic triphenyl phosphonium group. We observe poor colocalization inside the mitochondria. So lipophilic triphenyl phosphonium group, lipophilic cationic triphenyl phosphonium group is required to target mitochondria. So we have developed a rotaxane molecule where we have incorporated lipophilic cationic triphenyl phosphonium group to target live cell mitochondria. Squarene dye is required to image the mitochondria in the near infrared region. The macrocycle provides the center and protect the chromophore inside the mitochondria. Now question is how you can selectively target cancer cell over normal cell. So cancer is a worldwide problem, right? Cancer is the foremost threat for human life with higher mortality rate and for comorbidity. So this is the 2020 report. So almost 12 million people died due to cancer, whereas mortality of TB, malaria, and HIV almost remains constant. And this is the ICMR report 2025. So almost 30 million Indians may suffer from cancer. Now question is, if you want to target cancer, you have to understand chemistry and biology of the tumor cell. So what is biology of the tumor site? Endothelial gap in the tumor site is much more with respect to normal cell. It is known as leaky vasculature. It enhances the permeability of nanodrugs, and due to poor lymphatic drainage, it retains there. It is known as enhanced permeability and retention effect. A milestone discovery by Mayer in 1986. It is also known as passive targeting tumor. Another is active targeting tumor. Then you have to understand chemistry of the tumor site. So what is the chemistry of tumor site? So certain receptors are overexpressed at the tumor site, such as alpha b beta 3 integrin, epidermal growth factor receptor, folate receptor. So you can design suitable ligand to target those receptors. You can design RGD peptide to target alpha b beta 3 integrin. You can design folate derivative to target folate receptor. You can design suitable peptides to target epidermal growth factor receptor. So we have designed and constructed a rotaxane molecules where we have incorporated RGDS peptide, two RGDS peptide at the macrocycles to target alpha b beta 3 integrins which are overexpressed at the cancer cell. We have used squaring dye to image the compound in the near infrared region. We have incorporated morpholine functionality to target cellular lysosome. The macrocycle provide the shelter and protect the chromophore from nucleophilic attack inside the lysosome. So we have synthesized this rotaxane using solid phase synthesis protocol by using click chemistry. We have also synthesized a rotaxane, control rotaxane molecule lacking the morpholine group, which is lysosome targeting, right? Morpholine functionality. So we are lucky to have the crystal structures of the control dye, alkyne containing control dye, which also indicate that it is stabilized through donor-acceptor donor type interaction. And we have performed confocal laser scanning microscopic assay using epithelial surface carcinoma, HeLa cell line as well as lung adenocarcinoma, A549 cell line. And we have observed nice polarization inside the lysosome. And this is the video you can see lysosomal dynamics inside cancer cell by using our targeted rotaxane. And this is the multicolor imaging. The red dot indicates the lysosome. Our rotaxane based dye stain is lysosome, and this is the commercially available microcolor green for mitochondria, and this is the nucleus. So you can see in the cancer cell you can understand the crosstalk between lysosome with mitochondria, right? So this multicolor imaging is really important. You can see in a single cell you can see the cellular organelles. So for the control dye lacking the morpholine functionality, you observe poor colocalization inside the lysosome. So morpholine functionality is required to target cellular lysosome. So whether it is selective for cancerous cell or not, we have performed fluorescence activated cell sorting. So here we have observed 11-fold enhancement for cancerous cell compared to normal cell line. So we have designed and constructed a rotaxane where we have incorporated RGDS peptide residue to target alpha b beta 3 integrins which are overexpressed at the cancer cell. And morpholine is required to target cellular lysosome. Squaring is required to image the lysosome in the near infrared region, and macrocycle provide the shelter and protect the chromophore from nucleophilic attack. So the traditional single imaging method such as hospital radiography, ultrasonography, computed tomography, positron emission tomography, magnetic resonance imaging, SPECT have created huge interest in biomedical science and clinical diagnosis. Each imaging method has its inherent advantages and limitations in terms of penetration depth, sensitivity, image guided surgery capability, as well as cost. As well as risk of exposure to high energy ionizing radiation in the case of CT, as well as radioactive substances in the case of PET and SPECT. Right? Near infrared fluorescence has advantages. So you will get high sensitivity at the molecular level with fast feedback. Right? However, near infrared fluorescence has its inherent limitation. You will not get 3D tomographic information of soft tissues. On the other hand, as a non-invasive imaging technique, MRI possesses an unlimited penetration depth and provide anatomical 3D structural information of soft tissues. Right? However, MRI has its inherent limitation. You will not get molecular information. So, single imaging method is unlikely to meet all the criteria to resolve a precise diagnostic problem. So, you need multimodal imaging probe. So, multimodal several groups are working on the development of multimodal imaging probe. So, PET CT and PET MRI are already in use. Hospital দেখতে পাবে বা নার্সিং হোমে দেখতে পাবে PET CT PET MRI আছে. But the problem is that in case of CT you are using high energy ionizing radiation, and in case of PET and SPECT you are using radioactive substances. So integration of near infrared fluorescence with MRI is advantageous, right? Because you are not using high energy ionizing radiation, as well as you are not using what? Radioactive substances. But it is very difficult to integrate MRI with NRI fluorescence probe. Because often we have noticed that when fluorophore is attached to the superparamagnetic iron oxide nanoparticles, we have noticed the quenching of the fluorophore due to magnetic field. Now the question is how you will solve problem? How you will solve that problem? So here is Risopis, Sinarin, Luminem and Clariscan was previously used. FDA approved MRI agent. Now it is banned actually. Right? There are several problems of this. So we have designed and constructed iron oxide, superparamagnetic iron oxide nanoparticles, and it is rotaxane actually anchored on the superparamagnetic iron oxide nanoparticle surface. Right? Here we have incorporated lipophilic cationic triphenyl phosphonium group to target mitochondria. We have used DOPA, chemical functionality of DOPA, to anchor on the iron oxide nanoparticle surface. Squaring is required to image the mitochondria in the near infrared region. Their macrocycles provide the shelter and protect the chromophore from nucleophilic attack inside the mitochondria. Why we use carbohydrate? To enhance the solubility in water. Right? So that is why we have incorporated carbohydrate, particularly glucose. Glucose also have tendency to target cancer cells also. Right? And why we have used iron oxide Fe3O4? Because it acts as a superparamagnetic T2 agent. T2 content. In case of MRI, one is T1 contrast agent, gadolinium, 3+, manganese, these are T1 contrast agent, and iron oxide T2 contrast agent, right? So these are two different type of things. So we have particularly used T2 contrast agent here. So we have synthesized this squaring dye. This dye absorbs and emits in the near infrared region, but the problem is that this compound aggregates in presence of water. Now how you will solve this problem? So if you can interlock this dye inside a molecular container, then you can solve this problem. So that is why we have designed and synthesized these rotaxane molecules, and we have anchored this rotaxane molecule on the iron oxide nanoparticle surface, and we have synthesized this Fe3O4 nanoparticle starting from easily available chemicals FeCl3. And these are the characterization of these rotaxane cap iron oxide nanoparticles. It absorbs and emits, little bit broad, it absorbs and emits in the near infrared region with a fluorescence lifetime of 4 nanoseconds, and these are the characteristic IR. This part is for the iron oxide, and this part is for the rotaxane. Amide bond is there, right? And this is the high resolution TEM image. So you can see highly uniform structure, and this is the lattice fringes which indicate 83 nanoparticles. And this is the selected area for diffraction pattern with a diameter of around 15 nanometer. And we have performed field dependent magnetization curve, you can see, and which indicate superconductivity nature. And we have performed also confocal laser scanning microscopic assay using epithelial surface carcinoma error cell line as well as A549 cell line, and we have observed nice protocolization inside the mitochondria. And we have used MRI instrument, this MRI instrument. So you can see the darkening of the MRI along with the increment of the iron concentration indicated a good T2 weighted MRI contrast agent with a T2 relaxation rate of almost 181 millimole inverse second inverse, which is higher with respect to Faraday, Rizobis and scenario. The main problem of the confocal laser scanning microscopy is the resolution limit. Fluorescence microscopy main problem is resolution limit. 200 nanometer and नीचे जावा जाएगा. This is known as Abbe's diffraction limit. Now that is why in 2014 Professor Stephen Hell received the Nobel Prize for super resolution fluorescence microscopy. Now you can even go to nanometer, right? So he is the father of Stephen Hell. He is the father of super resolution microscopy. But the problem is that you have to use a excitation laser and you have to use a state definition laser and high energy state definition laser. And almost all the compounds bleaches. And you don't have any targeted, so far you don't have any targeted probe using state microscope. So that is why we have designed and constructed rotation based probe. So here you can see this is the confocal image. It is not possible to resolve crystal structure. Distance between two crystals is around 80 nanometer. So it is not possible by using confocal microscopy to resolve that crystal structure. But we did by using state microscopy. Here you can see we can resolve the crystal structure inside the mind. So this is the first report of rotation based probe using state microscopy. And you can see this is the mitochondrial dynamics using state microscopy. So you can see the mitochondrial fusion and fission. Mitochondria is very important. Mitochondria যদি খারাপ হয়ে যায় আইদার লাইসোজোমটাকে খেয়ে নেয় অথবা আরেকটা মাইটোকন্ড্রিয়া কি হয় ফিউজ করে ওটাকে বাঁচিয়ে দিয়ে আবার কি করে ভেঙে দেয়। এটা খুব ইম্পর্টেন্ট in mitochondria. So mitochondrial dynamics is really important. So this one is state microscopy. We can see even also the crystal structure, but this one is also confocal microscopy. It is not possible to resolve this crystal structure also. So we have designed and constructed a rotation molecule. This is the first reported rotation molecule utilizing state microscopy. So now our group is working on the development of rotaxanes, particularly mitochondria targeted rotaxanes. So whether we can control cellular function or fate. So this is our ongoing project. So in conclusion, I must say, so we have designed and constructed rotaxane molecules using solid phase synthesis protocol. We have synthesized some mitochondria targeting as well as lysosome targeting rotaxanes. We have designed multimodal imaging probes integrating with near infrared fluorescence with MRI, and this type of probe, this because this is magnetic material, right? So you can do continuous magnetization and demagnetization to develop hyperthermia. So currently this type of work is also going in our lab. So we have developed rotaxane based state probe, targeted state probe, Guha targeted state probe. So now If I ask you what is machine, what you will say? See the clipping from 3 idiots.
What is your machine?
He asked what is machine? वो हास्यल वो एबार आंसर दे ये पाका भरे मेশিন টিভি চলছে মেশিন মুখস্থ করে আসেন আর ও বলছে তো আমি বুঝতে জানি এগুলো আমার মুখস্থ ছিল So, this is why ego is trying to learn the topic actually. Don't just only memorize things. So, at the end I must say, this is the I learned that biology, physics, everybody will be here, but I think only chemistry or what. So, I must know this is the rise of in physics in 1944 for NMR. So I quote, my mother made me become a scientist without ever intending to. Every other Jewish mother in Brooklyn asked her child after school, did you learn anything today? Not my mother is it. She always said, did you ask a good question? That difference. Asking good question made me become a scientist. So I must quote, if you can't explain it simply, you don't understand it well enough, Albert Einstein. The important thing is to never stop questioning. So, this is my small group. They are the architect. I did nothing. They are the architect of this project. Ravi, Shamiran, Pranab, Taposh, Payan, Snehako, Sucharita, Ipshita, Priti, Aprili. They are the architects of this project. They design all the work and they are working this. Right? So, I must acknowledge ACRB and National Research Foundation for the financial support as well as DSCBT, Government of West Bengal and Jadavpur University for the financial support. And they are our collaborators. So thank you very much for your kind attention.
ধন্যবাদ স্যারকে। আজকে আমরা আমাদের দ্বিতীয় দিনে স্যারকে পেয়ে আমরা অত্যন্ত খুশি হয়েছি। ওনার পড়ানোর ধরন মানে শুরু থেকে শেষ অবধি আমরা যতটুকু বুঝতে পেরেছি সত্যি আমাদের কাছে অনেকটাই পাওয়া। স্যারকে একটাই অনুরোধ, সেটা হচ্ছে যে আগামী দিনেও আমরা যখন এরকম ইনিশিয়েটিভ করবো আপনি অবশ্যই আমাদের কারণ আপনার গাইডেন্স ছাড়া আপনাদের গাইডেন্স ছাড়া আমরা কতটা করতে পারবো না। ভেরি মাচ ফর ইউর ইনভাইটেশন এন্ড এভরিথিং। থ্যাংক ইউ স্যার। Thank you very much.